Logical theme. All right, so I'll ask you a question. Boy, right? We left off, we were about to talk about proteins. I want to talk about the structure of proteins. I want you to tell me what makes up the primary structure of a protein. What makes up the primary structure of a protein? You guys should all have Pear Deck up, not your vocab. Primary structure of a protein. I need the Jeopardy music. It is. Just be happy it's not like Ukraine and Putin shutting off the natural gas, right?
I could pull it up for you. I still like the foot picture that we had Mr. Kaiser and Joseph.
See, we can learn history and biology all at the same time. I can get it. All right, here we go. Let's see what we end up with. Okay, you told me the primary structure is the amino acids, carbon. Oh, I like that one. It's my favorite one. The order of amino acids in the chain. Very good. The order of two of you had it perfectly right. The order of amino acids is that primary structure. So the order you put your amino acids in gives you the primary, that very first structural component of a polypeptide or protein. Okay? Remember, proteins fold, and then you get the secondary, tertiary, and quaternary structure. We'll talk more about that. So what do these proteins do? The best answer to that is everything, right? So we have over 30,000 genes in our body, and they all code for a protein, right? Or a polypeptide, I should say, that might fold into a protein later. But because we have all of these different proteins, and they are basically what our goal of our metabolism is to make more of them. That's why our cells work and function. That's why DNA exists. That's why we have fats to store energy. That's why we have carbohydrates to feed this metabolism and this production of proteins. It all comes down to making these functional structures called proteins. So we know the metabolism, and most enzymes are proteins that act as catalysts to accelerate chemical reactions within the cells. We all remember metabolism is the sum of all chemical reactions in the body, right? Everything you build up and everything you break down. All of the catabolism and all of the anabolism together make up your metabolism. Enzymes make it so that you don't have to light a fire to break down a steak in your stomach. Imagine the amount of energy you would need to turn a pile of meat into ash and break it down to its simplest components. That happens in your tummy, right? And we do that with enzymes. Instead of needing that fire, we can reduce the amount of energy for a reaction to occur using an enzyme to lower activation energy, right? Does that make sense? So looking at support, how do we have proteins that support us? Keratin. I want you to point at your neighbor, and I want you to look down at your finger and say, "You're looking at my keratin." Say it to your neighbor. Yes, you work with. Oh, yes, you are a keratin expert. Yes. I don't. I have it. I'm looking for a joke, and it's not there, but I'm going to have one by the end of the hour. So, and then we have collagen. Anybody ever heard of collagen before? It's what bones and what all of your cartilage are made of, right? It's that structural component. Glucosamine and chondroitin. Who's heard of that one? It's a supplement people take for their knees. There's also collagen in there that they have you take. How about this one? Here's something really gross about collagen. You ready for this one? Who has ate Jell-O? Yeah, Jell-O, right? Okay. So Jell-O, what makes Jell-O jiggle? Cow bones, right? They basically boil a big giant vat of cow bones until it's like goo. And then they dry it, and then they grind it into powder, and they put it in these little pouches called gelatin, and then they sell that to you, and you mix that up, and it thickens, right? It gives you structure to your Jell-O. Essentially does the same thing in cartilage and bones, right? It gives it structure. Okay? That's a protein. Collagen. Then we have different types of... Am I giving you illustrative examples as we talk about these, by the way? Okay, all right. So keep that in mind. That's why I'm jibber-jabbering so much. Okay? So looking at transport, we have hemoglobin. Hemoglobin, we know, is the protein that sets on top of a red blood cell and carries oxygen. Pretty important, right? And then we have membrane proteins. Some of them just hold up signs that say, hello, I'm supposed to be here, don't kill me, right? Or I belong here, right? It's like name tags, okay? Right? We talked about those glycoproteins, okay? Then we have defense proteins. These are like antibodies. Some of them are antigens. Sometimes antigens' job are to walk around like let's say I had a pile of Post-its and I just walk around and I go, oh, you're not supposed to be here, and I tag you, and then I tag you, and then a white blood cell comes around and does what? Kills you because you weren't supposed to be there, okay? So that is how antigen proteins work. They can literally tag a cell with a protein. They're called beta cells, and then the big killer T cells combine and do the big destroying, okay? So next one is regulation. These are like hormones, right? They help regulate things. What's a good example I can think of here? You get hungry, your body stops releasing what's called serotonin. That's a pretty big deal. It makes you really what? Cranky, unhappy, right? Okay. Anybody get cranky when they're hungry? Oh, I'm right there. I'm like, if I haven't ate and my body stops giving me serotonin, I look around and I'm like, we need to kill something and eat it, like right now. I don't care what it is. Yes? Well, being around people that you love and like does help you release serotonin too. So if you're really hungry, and you're next to your worst enemy, or someone's picking on you, it's going to make things like really bad. You're more apt to snap out and lash out at them. Whereas if you're, like, let's say I was really hungry and I'm next to, my wife, who I love, we're probably not going to snap at her. We're probably going to get along and be like, it's okay, we're going to eat soon. We'll get there eventually. You get the idea, right? And if you release serotonin, then you get to release this other thing called dopamine in your brain, which makes you happy. Drugs do the same thing, except it's like drugs are called dope, except they do it too much, right? Okay. So then we have motion. Let's move on here. Muscle proteins, actin and myosin. You guys, we're going to talk about these later. You might even want to write them down. Actin and myosin are the two proteins that are involved in muscle contraction. Actin, A-C-T-I-N, and myosin, M-Y-O-S-I-N. So those two allow you to move. Pretty important. Now those are just like literally 6 examples of sort of the 30,000 we have, right? So, and then there's other organisms which have different sets of proteins. So that's just our proteins. We should have a class just called proteins, right? There is proteomics. There's a study of proteins. So looking at amino acids and how they're built, there's 20 different common amino acids. And amino acids differ by what's called their R group. What's an R group? What? Functional group. Yep, functional group. Okay. So if you look at this molecule and you go, okay, well, that's not R, that's not R, that's not R, that's not R, that's an R. That means all amino acids are going to have what's called an amino group. They're going to have a carboxyl group. And then they're going to have a hydrogen. And they're going to have a central carbon. And then this one area, this one bond on this carbon is going to change. It's called the R group or the functional group. There's 20 different R groups for amino acids. Therefore, there are 20 different amino acids. Yes, sir?
So besides the R group, are all proteins going to look like this?
This is an amino acid, not a protein. But all amino acids will have these 4 things in common: a central carbon, carboxyl, hydrogen, and an amino group. The only thing that will be different is this. Okay? So looking at some of the different examples of that, you can see all of these amino acids, and I don't care which one I am. You can, how come it's just blocked off for some reason. Anyway, they all have that carboxyl group, a hydrogen, an amino group, amino, hydrogen, carboxyl, and you can see the thing that differs is indeed that functional group. And every time it changes, I change into a completely different amino acid. Does that make sense? Do you think if I gave you any one of these, you could identify that there's an amino, a hydrogen, carboxyl group to a central carbon, and then there's something different over here? Yeah. I mean, if you can pull these simple concepts out of a problem on an AP exam, you're going to be fine in this category. So again, now we have to look at how we would put these together, because to get a polypeptide or just a dipeptide, I have to have a way that they're going to bond. What type of reaction, let me ask you a question, what type of reaction is going to bring two amino acids together? What type of reaction will bring two amino acids together? What type of reaction will bring two amino acids together? Dun, 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 dun. Give you 10 more seconds. 9, 8, 7, 4, 2, 1. Let's see what we did. Physical peptide. Together, not hydrolysis. There's a dehydration reaction. I like that. There we go. It is a peptide bond, but through dehydration synthesis, you should have had perfectly, you're going to make a dipeptide. So I'm glad I asked that question, because that's something we need to take out. When we're bringing these biomolecules together, understand, together means synthesis, right? So if you hear the word together, you know it's some type of dehydration synthesis. If it's apart, it's probably hydrolysis, right? Hydrolysis. So let's just look at these. I have what here? Yeah, if I take that off and I put it with one of these and carbon bonds to nitrogen, I might have a success putting these two together, right? And I might make what in the process? Water, right? Okay. So if I add those two together through dehydration synthesis by bonding a hydrogen and a hydroxyl group, I'm going to create water, therefore bonding my molecules together. Does that make sense? Oops, sorry. Are there any questions on this basic concept of dehydration synthesis and hydrolysis? By the way, that's one of the key things in this chapter. Okay, you're going to see that on the AP exam, promise. Okay. 1, I don't know if it's going to be hydrolysis. I don't know if it's going to be a dehydration synthesis. I don't know if it's going to be both, but you're going to see it on there. Yes? Yes, sir. Yep, that's the only way they bond. Otherwise, I'd have disorder, right? It'd be chaos. Amino acids would be flipped upside down. And, yeah. Good question, though. So levels of protein structures. That's why I say, you know, life's really simple when you look at it. I mean, I know organic chemistry is hard, but the organic chemistry that makes up life is not that difficult. Once you learn it, the rules stay the same. If life didn't play by the rules, it would be too complicated to exist. And it would never have evolved to be here in the first place. So proteins cannot function properly unless they fold into their proper shape. Now, when a protein loses its proper shape, it's said to be denatured. So we said we can use proteins in enzymes over and over again, right? Unless they become what? Denatured. Then we got a problem. Then things don't fit. Now I have a problem with my structure, therefore I have a problem with my function. So exposure of proteins to certain chemicals, like a change in pH or a high temperature, can disrupt protein structure. Now, some of you did some pretty neat experiments in your seeds. Some of you stuck things in a microwave. Do you think that could change the protein structure of the seed? It could, right? Some of you are going to adjust the pH. Do you think that could mess with proteins? It could. So I'm not saying that's why, but I'm saying that could be part of why, right? We don't know. So proteins can have up to 4 levels of structure. I asked you about the primary at the beginning of the hour. We all know now that's the order of amino acids. But then it's going to start to fold and develop different structural units. And that's where the secondary, tertiary, and quaternary structure of a protein take place. If I don't get my final quaternary structure, I don't function. I'm not considered a protein. I'm still a polypeptide, right? Okay. So the first primary structure is the sequence of amino acids. Very simply put, the order you put the amino acids that are present in that polypeptide in. The secondary shape is characterized by the presence of alpha helices and beta sheets held in place with hydrogen bonds. Let me put that in English. When that protein starts to fold, because imagine a pile of string and you dropping it on the table. And now imagine some of those amino acids carry a partial charge, right? Do you think that just like water, they might bond to themselves a little bit with hydrogen bonding? Yes. So this first structure are those initial hydrogen bonds that take place between some of the amino acids in that polypeptide. Thirdly, the final overall three-dimensional shape. Now I'm talking about continuing to fold, right? Let's say I now pick up that pile off the table and I put it in a ball in my hand. More bonds are going to take place, okay? Stabilized by the presence of hydrophobic interactions. I just added a component, didn't I? Now I'm not talking about hydrogen bonding between the amino acids. Tertiary structures, proteins are going to be submerged in a solvent called what? Water. So that means some of the amino acids are going to repel water, some are going to be attracted to it. Because of that, I have this tertiary structure where I'm going to have different bonding taking place, and that's going to force different parts of my proteins to bond to each other as well. It's actually going to force covalent bonds to take place because certain portions of the amino acids are going to be repelled so much that they'll break bonds within that protein structure. That's the tertiary structure. The quaternary structure sometimes does not exist. But if I combine two polypeptides, which often you do, to make a structural protein, now I'm looking at the final stage. The tertiary structure can function as a protein. The quaternary structure is if you bring two different polypeptides or more together to form a functional, what we call a protein in the end. Okay? So often you have polypeptides that have a tertiary structure that act as a protein, and then you have quaternary structures which involve more than one polypeptide coming together. Okay? Now that's a way more complex definition than I give you in biology. I know that. Okay, but that's why we're here, right? So now we understand that there's way more involved in the folding of a protein to get to that quaternary structure in functional protein. So what does that look like? Now, this is just a caricature, obviously, but we have that primary structure, just that order of amino acids. Then we have the secondary structure where you can start to see some of the hydrogen bonding taking place between the amino acids of adjacent amino acids. Then, if I start to fold, and through being afraid of or scared of water or attracted to water, I'm going to start to fold loops and more folds. Basically, these molecules either running or being attracted or pulled towards water. Proteins literally look like a mess. But when you see them function, they do their job perfectly. Okay? Remember, evolution doesn't have to be perfect. It just has to what? Work, right? And this does. And then that final quaternary structure, you can see here how there's two different colors representing two different protein structures combining. This is very much like a ribosome. Now, ribosomes made of proteins, but I look at that as a larger version of a protein. A ribosome has two parts, right, that fold together on top of mRNA. And that's why I say it's kind of like one in a way, if that makes sense to you. Proteins function like that a lot. Sometimes there's two polypeptides that come together to form a unit that then function as a protein. That would be my quaternary structure. What questions do you have about proteins? Yes? The secondary right here? That's when the parts of the amino acid chain are repelled to or attracted to water because it's submerged in water, and that's going to start to create that secondary structure. The tertiary structure is when all of that repulsion actually causes parts of it to bond to other parts, and now we're forming like new covalent bonds, new ionic bonds. And then the quaternary is when two proteins are together, or polypeptides, sorry, are together. So looking at different protein folding diseases, sometimes folding doesn't happen the way it's supposed to. And if I don't fold correctly, I get a bad structure. If I get a bad structure, I get a bad function. So chaperone proteins are going to help proteins fold into their normal shape. So sometimes we have proteins that guide the folding. And defects in chaperone proteins may play a role in several human diseases like Alzheimer's disease and cystic fibrosis. So cystic fibrosis, I'll talk about that really quickly. There's a chaperone protein that helps my lung tissue, the proteins in my lungs, fold correctly. If I get a malfolding in my lung tissue and it's not pliable and flexible and it becomes very rigid because of my bad fold, It's like bad origami. Anybody ever seen bad origami? If you've seen me do it. Yeah, anytime you try. Okay, it doesn't look like it functions very well, right? And it doesn't. Just like if you fold a protein in your lung, it won't function like lung tissue. If lung tissue doesn't expand and contract very smoothly and very pliably, you can't expel all the fluid from your lungs, and it's a problem. People with cystic fibrosis usually die before the age of 30. Okay? Little kids, you ever see anybody having to pound their kid's back with cystic fibrosis, right? That's because they're trying to help the lungs knock all that fluid out. Because usually they die of like pneumonia. Okay? So that's just one case of a disease from the malfolding. And that would be a chaperone protein that's supposed to be the folder, so to speak. Okay? That'd be like me doing origami. It's not going to look so good. So prions are misfolded proteins that have been implicated in a group of fatal brain diseases known as TSEs. These prions are known to cause brain tumors, essentially. Now, mad cow disease is one example of TSE disease. We've all heard of mad cow disease, right? That's where you get a malfunctioning protein that's not folding the proteins in your brain right. That sounds pretty big, right? Okay. That means, yeah, big problem. It's like cystic fibrosis of the brain in a way. Okay. The bad part about that is, if you get those prions in you, guess what? Yeah, you can end up like the cow. Yeah. Finally, my favorite group, the nucleic acids, right? I love my DNA. Who doesn't? Makes all my proteins. Kind of. So nucleic acids are polymers of nucleotides. There's two varieties of these nucleotides. There's DNA and RNA. There's actually three. We'll talk about the other one later. But DNA we know is deoxyribonucleic acid, named after its sugar. Genetic material that stores information for its own replication and for the sequencing of amino acids and proteins. Remember, we copy DNA onto RNA. RNA wiggles through a nuclear pore, goes out into the cytoplasm to assemble amino acids at a ribosome. Okay? So that means there's this other thing called RNA. Now, DNA is this very large structure trapped inside the nucleus, right? Can't get out. It's double-stranded. For us, it's 30,000 genes long. Can't fit through a nuclear pore, too big. It's literally 60,000 times bigger than one RNA strand. Okay? So we copy DNA again onto RNA, and this performs a wide range of functions within cells, which include protein synthesis and regulation of gene expression. If I say gene expression, I'm talking about transcription and translation. Now, we're going to spend a chapter on that later. But if you've had me, you understand gene expression is copying DNA onto RNA and the whole process out to a ribosome to make a protein. How many types of RNA are there? Oh, this is a trick question. 3 basic types. You're right in that. Well, I'll tell you about a few more this year. Remember, there's mRNA, tRNA, and rRNA. There's also a few others. So each nucleotide is composed of 3 basic parts. A phosphate group, see phosphate. What do we know about phosphate group? It's going to attract oxygen, which is electronegative. So if you have a phosphate, that part of you is going to be negative, right? Okay. So it also contains a pentose sugar. Pentose means how many carbons? 5, right? So we know nucleotides are going to have a 5-carbon sugar and a phosphate. Makes sense? And then they're going to have this other nitrogen-containing base. Now we know that there's 5 types of nucleotides found in nucleic acids. We know DNA contains adenine, guanine, cytosine, and thymine. And we know that adenine, guanine, cytosine, and uracil instead of thymine are found in RNA. This is basic DNA knowledge from last year or the year before. And we understand that nucleotides are joined together by a series of dehydration synthesis reactions to form a linear molecule called a strand. So when I say strand, I'm referring to basically a chain of these, right? I know we have a while. I was just checking the time. Couldn't remember exactly what it was, 49. You can get out of here, yep. Just make sure to study for the vocab test tomorrow. Okay. Let me ask, let's do this real quick. How about, I'm going to give you a DNA strand. Please give me the RNA strand that would go with this DNA strand. Please give me the RNA strand that would go with this DNA strand. Good luck. I hope that you're golfing, right? I mean, if I could leave and go golf right now, I wouldn't because I just love being here. Right? Yes, please. I want RNA. Oh, I need to go back? Okay. Hold on. GC Tata. All right. So I'm helping you remember it. All right, let's see what we ended up with. Ooh, I don't have the same ones. Ooh, I got things all over the place. Okay. So I said CG Tata. So that should have been G, C, and then T gets replaced with a U, but I asked for the corresponding one, right? Let's do the, I'll do it right over top of this here. So I said GC Tata. You should have wrote as the corresponding mRNA CG. T still goes with A, right? A goes with U now. T goes with A, and A we know goes with U. So you should have had CGAUAU. So who had that? CGAUAU. CGAUAU, there's one. Okay. So hopefully you understand AU, not UA. Okay. So the problem what you were doing is you were just replacing the T with a U. You needed to do the actual transcription process, okay? Don't worry if that's still a little foreign to you. We are going to cover that in a future chapter, I promise. So if I look at the monomers, the basic monomers of nucleic acids, it's a nucleotide. We understand it's a nucleotide, okay? Those are going to contain my 5-pentose sugar, which has that oxygen. Look at that oxygen. No, that's the wrong oxygen. I'm sorry. Look at that phosphate group. What do I know is attached to that phosphate group? 3 oxygens, right? Okay? And if I have all those oxygens, I know it's electronegative. So we know that this phosphate group right here is going to have a little negative polarity to it, which is going to make this whole group attracted to water. Okay? We'll talk more about that later. I know a lot of you have already done a DNA extraction with me. But the 5-pentose sugar, the phosphate group, those will always be the same, but this nitrogen base will change. So here, why do I say pentose sugar? Why don't I say deoxyribose or ribose? I'm generally referring to nucleic acids and the nucleic acids in general. So this pentose sugar could be either ribose or deoxyribose. Deoxyribose, it's a DNA nucleotide. Ribose, it's an RNA nucleotide. Okay? So this phosphate group has something very special going on, which I've mentioned over and over again. And this is like one underlining thing that often is not stressed upon, that you'll miss test questions over. You need to understand that because of that phosphate is there and is attracted to so many of these oxygens, oxygen is electronegative, more electronegative than most. It is going to draw a slightly negative charge, which is why I can actually do a DNA fingerprint. I can actually drag DNA across a median by its charge. And we'll do that a little bit in here this year, okay? So if you didn't have biotech, don't worry. We'll get to do a little bit of DNA fingerprinting this year. Okay? All right, so that nitrogen base will shift and will change, but the thing that will change as well is going to be the sugar. Deoxyribose is the sugar in DNA, go figure, deoxyribonucleic acid, and ribose is the sugar in RNA, go figure, it's ribonucleic acid. So that pentose sugar will shift and change. The other thing you need to be able to identify are the different types of nucleotides and the different types of nitrogen bases. So we understand that there are pyrimidines and there are purines. Pyrimidines are going to be our single carbon ring bases, whereas purines are our double carbon ring bases. The way I remember this is this simple. I always remember pure silver kills werewolves. Pure purines is Ag. Silver Ag, right? Okay? I don't care how you remember it. That's how I remember it. And if I know Ag, I automatically know the other ones are pyrimidines, right? So CT and U, I don't have a fancy way of remembering that. No. What is it? Tuck? What? Cut? Oh, yeah, cut. Yeah, like the Mayans cut the heads off on top of the pyramids. Kind of, but it wasn't the pyramids though. I know. I see. Did I tell you guys I was helping my kids with the Mayans and the Aztecs? I wrecked my daughter's world when I told her that Mayans did human sacrifice, because that was not in the book. I was like, you know what they used to do, and she's like, they did not, and then she went and looked it up. Anyway, not a big fan of social studies. What a boring topic. So wrong. Anyway, so if you take a look here, please know the three different types of pyrimidines. Understand that cytosine and thymine are found in what molecule? DNA, whereas we replace thymine with uracil, so I have C and U in RNA. That's a dead giveaway. If you see something with a T in it, you know it's DNA. If you see something with a U in it, you know it's RNA. Sometimes that's the slightest thing that you're given on a test, and you better be able to identify that. So looking at the structure of DNA and RNA, the backbone of the nucleic acid strand is composed of alternating sugar phosphate molecules, and RNA is predominantly a single-stranded molecule, whereas DNA is a double-stranded molecule. I say RNA is predominantly because sometimes RNA folds on itself and parts of it become double-stranded, like in tRNA, right? You guys remember tRNA? Okay. We'll talk more about those, don't worry. But looking at DNA, it's composed of two strands held together by hydrogen bonds in the middle. Hydrogen bonds strong or weak? Relatively, well, they're stronger than ionic, but they're relatively weak if I'm looking at a biomolecule, right? So the nitrogen-containing bases in the middle are where those weak hydrogen bonds will take place, and the two strands twist around each other to form a double helix. The side chains are covalent bonds between the sugars and phosphates of adjacent nucleotides. So adenine hydrogen bonds with thymine, cytosine hydrogen bonds with guanine. You always have a pyrimidine with a purine. If I had two purines next to each other, it'd be two double carbon rings. They wouldn't bond. They wouldn't mix together. It'd be like a bubble in my DNA. If I had two pyrimidines together, it'd be like a divot in my DNA, and they would repel each other anyway. They wouldn't bond. Okay? So the bonding between the nucleotides in DNA is referred to as what's called complementary base pairing. We call it the complementary base pairing rules. So looking at a basic RNA structure, we understand that we have phosphates, we have our pentose sugar, and the pentose sugar in this case is what? Ribose, and then we have our different nitrogen bases. We understand that we have cytosine, we understand that we have adenine, we understand we have uracil instead of thymine, and then we also have guanine, okay? So it's similar to DNA nucleotides, but the structure it makes is extremely short, it's only single-stranded, and it has a different sugar, and it has uracil instead of thymine. Quite a lot of, I wish they'd draw these a lot different, but they do look similar as far as a single strand, but they are very, very different than DNA when you really look at them. We're talking about different sugars, at least one different nitrogen base, very short, single-stranded. And then you have this DNA, this giant, Giganosaurus molecule. Isn't that the new dinosaur they just found? Is that what they called it? Yeah. You know what I wish they would have called it? The new dinosaur. Why didn't they just call it Godzilla? That would have been awesome. Yeah. Yep. I wish they would have called it I'm like, why? In my head, I'm like, why didn't that scientist just call that thing Godzilla? I found Godzilla. You could say that, you know. No. Trying to wake you guys up with a little bit of bad humor. It's not working. I know. I hear you. So they're going to think that I understand. Okay. So DNA, very similar, except very different, right? DNA is double-stranded. It has the same phosphate group, a different pentose sugar. This time it has deoxyribose. It's going to have cytosine bonding with guanine and adenine bonding with thymine. It does not have uracil. So how do you remember the base pairing rules? A goes with T and G goes with C. I always remember this. I go at AT, G and C to get my vitamins. I always remember that, and that's how I put it in my head. So A with T, G with C. Or you can remember G and C both have a curve in them. They go together. I don't care how you remember it, but as long as you remember it. Okay. You know, A and a T both have horizontal lines, you know. I don't know. Maybe. Well, then you got the G with the, I don't know. That might not work. Isn't that weird how different people's brains work? Yeah. I think so too. I remember things so oddly sometimes. So looking at the differences and similarities, you should know that the sugar in DNA is deoxyribose. The sugar in RNA is ribose. The bases in DNA are adenine, guanine, thymine, and cytosine. In RNA, they're a little bit different. We have uracil instead of thymine. The DNA we know is double-stranded with base pairings, and RNA is single-stranded. You should also know that RNA is super-duper short compared to DNA. Okay, that's huge. Our DNA is 30,000 base pairs long. RNA is one of those, right? Just think about that. And then it's half of a strand. So it's really 1/60,000th the size of our DNA. RNA is. Just the size difference is huge. Okay. Not to mention RNA splicing, which we haven't even talked about yet. It's actually probably 1/220,000th the size. So there is a special nucleotide. This is the one I don't ever really tell you is a nucleotide in biology, but I need to have you reference it as a nucleotide in here. There is. It is not. I repeat, it is not a nucleic acid, but it does fall under the nucleotide, the monomer category. It never goes beyond this stage. It does not become a macromolecule or a polymer. It is ATP, adenosine triphosphate. So this is composed of adenine, ribose, and 3 phosphates. It is literally named after its components. Adenine, adeno, ribose, the sugar, adenose, right? Tri, 3 phosphate. So adenosine triphosphate is what it's named, how it's named. So ATP is a high energy molecule due to the presence of the last 2 unstable phosphate bonds. Phosphates are really attracted to what? We've been talking about this. They are, but because of the electronegative oxygens, right? Okay, so because they have that electronegativity on their end, and they've got 3 of these phosphates. Electronegativity makes you very attracted to other things, right? And they got 3 of them. So they're going to be highly reactive, slightly, right? Just think about it. I say that because they don't just break down on their own, okay? So hydrolysis of that last phosphate on the end is going, now I said hydrolysis, right? So what am I going to do in hydrolysis? Break it down using water, okay? And there's water in our body, right? So if I provide enough energy, I can actually break a bond, releasing even more energy in this process. So the molecule of ADP, adenosine diphosphate, will be produced, and we get an inorganic phosphate released, okay? That can reattach later if I store energy. Energy to do cellular work is harvested this way. This is how we get our energy for our cells to do their actual chemical reactions. And ATP is called the energy currency of the cell. This is how we transfer, store, and use energy in our bodies. This is how plants do it as well. This is no secret if you've had me for biology. So this is what ATP kind of looks like in a model, all right? Look at my chipmunk. Oh, he's so cute. All that energy in his cheeks. Look at that. Bunch of ATP waiting to be released. All right, so we have adenosine. We see our pentose sugar, and over here we see adenine. Adenine is a what? A nitrogen base, more specifically a purine. I like the way you guys are thinking. So here I have my sugar, my pentose sugar, my ribose, right? And I have my adenine, together adenosine. And then I have a phosphate group. But instead of one, I have how many? 3. That makes me this energy carrier, right? So if I add water to this, if I need energy, I can break it down, okay, releasing a phosphate and free energy becoming ADP. I gave you a spot to draw, I believe. Okay. So I have adenine, which is going to be bonded to that first phosphate covalently. This will not break. You're going to draw this in your notes. There's a spot to draw in your actual notes. I'm sorry, not on Pear Deck. So then I have these 2 other phosphate groups, and most biologists, not necessarily chemists, will draw these high energy bonds this way. Okay? It's like a biological way to draw special bonds that will break. Does biology break a lot of rules sometimes? Yeah, that's okay. So if I break this bond, I release what? Energy and also a phosphate. And ATP becomes ADP. Now, in a starvation situation, do I still have energy I could break off here? Yes. I could break this off, release even more energy, and release a phosphate, and then I would become what's called AMP, adenosine monophosphate. I can't break this down anymore. That will not normally happen. This AMP will not normally happen. So this is AMP, this is ADP, this is ATP, also where Native Americans live. Okay. Halloween's coming. I expect one of these in Mr. Urick's yard. TP. Okay. Anyway. Oh, you got evidence of me saying that now. It's not good. Yeah. Yeah, kids got Mr. Urick last year. I don't know how they got his address. That was pretty creepy. So, if I'm going this way, this is going to be obviously kinetic energy, right? But what reaction is occurring? I'm going to ask you in Pear Deck. No, wait. What reaction is occurring if I go down? Hydrolysis. What if I go up and store energy? It must be dehydration synthesis. You have a vocab test tomorrow. Vocab test tomorrow. Everything else can wait till tomorrow.